I would now like to welcome our next speaker to the floor, Murat Tunaboylu, co-founder and CEO at Antiverse, who will be speaking about AI-augmented antibody drug development and discovery. Murat, it's a pleasure to have you with us. The floor is yours.
Hi, everyone. My name is Murat Tunaboylu. I'm the CEO and co-founder of Antiverse. We are here to engineer the future of drug discovery. And when we say that, we are mostly talking about biology. I would like to give a little bit of a context before I go into my presentation. We found Antiverse with my co-founder Ben Holland back in 2017 with the idea that rather than discovering drugs and antibodies, we can maybe design them. However, the interactions between antibodies and targets are not very well known. So then the idea was to be able to generate a lot of data, and by looking at data, infer the relationship between antigens and antibodies. And then once establishing that relationship, the plan is to be able to use that information on new targets and coming up with a list of new designs, biologics designs, which to be then expressed and tested in the lab. So that's been the goal of the company from very early on. In 2017, April, we had our first proof of concept, got the initial accelerated based funding. And since then, we basically repeated incrementally more difficult proof of concept projects. And 5 years later, we are a company, an AI first drug discovery company that raised about £2 million. We are a team of 8 with advisors under 20. We are based in the UK. Our lab and office is in Cardiff. We are post-revenue. We work with some top 20 pharma, CROs, biotechs. And the idea is to help them design biologics better, faster, against any given target, and especially challenging. We have 2 types of projects. One of them is called AI-augmented drug discovery. I'll get into the details of this, but the benefit of this platform to the users is that they have more diverse, rare, unique antibody sequences as part of their search. This sits on top of their drug discovery process rather than replacing it, hence the name augmentation. Our partners found 10 times more diverse antibodies through this collaboration. We helped them to use the full potential of the library for the antibody libraries they spent so much time and effort on. The second thing we do, we can do the whole thing right now. So a partner points to a particularly challenging target, a GPCR in many cases, and then we take it from there and provide them a functional antibody 6 months later. That involves using stable cell lines, designing target-specific antibody libraries. After synthesizing those libraries and putting into phage, we do the screening in-house. And by studying the results of that panning, we can look for unique antibody clusters and the best candidates from those clusters, and then test them, and then finally run functional tests after that. And for GPCRs, the most common things we do is the C-AMP and the beta-arrestin. So I'll take a moment and then talk a little bit more about the service. The AI augmented service that we offer corrects for 2 problems. One of them is what we call as amplification bias. So once you design a library and have a platform to display that library, pan that library against antigen and do amplification, at that stage, we are selecting for ease of expression rather than strength of binding. And even 4% of slowness in expression can cause a quicker expressing sequences to overtake the whole population or become very dominant. So selecting for expression rather than selecting for binding strength. The second problem is most common, or the caveat of this type of process. People look at a few hundred clones from the final pen with the hope to find very diverse antibodies. Well, that search can give you some antibodies, maybe some of them different as well, But time after time, we saw that you only hit the most dominant sequences and miss those rare and unique antibody sequences. Both of the problems you can fix for. And our suggestion to that is basically sequencing every pan, getting those sequencing in, putting on top of our proprietary antigen antibody database, and study them there, and then cluster those antibody sequences, not only based on sequence homology, but also for 100 other characteristics, including their structure. Now, once we have those clusters, we select the most representative sequences from each cluster, put into a list, and tell to our partner that, look, on top of the 10 antibody sequences that you found, here's another 90 antibody sequences that are all diverse and have different characteristics. And this way, maybe you can explore the full functional space and cover as many epitopes as you can with the same testing budget. So this is how it works out. A partner comes to us, they say that, look, they have an amazing library. They did panning against a target, but it didn't result antibodies that they would like to necessarily continue with, or they found someone's, but they know that there is more inside the library, and they ask for our help to untap that potential. So what we tell them, that's great. Please send the output of each panning as a PCR product to an NGS facility, and then we get the heavy and light pairs separately and combine internally, or if it's a nanobody, it's heavy only. So once we have the data, we do our machine learning algorithm-based clustering and antibody selection, which takes a few weeks. And in particular cases where there is one cluster that seems to be very promising, which doesn't have enough sequences, we generate new sequences in that group. We collate our results, we give it back to our partner, and our partner then can either recover those sequences from the library, if they are coming from the library, with a two-step PCR process, or by using the full sequence, they can synthesize and test them. So we have done this already many times with different, in commercial context with many companies, and found results where you have 6 to 13 times more diverse output. And some of them cross-reactive antibodies against the murine homologs. In other cases where you find different mechanisms. The thing is, this is a very standard thing that now we have developed. We developed originally internally for our use, but then after seeing a lot of interest, we package and offering as a service for fee for service business model. But that's not the only thing we do. As we would like to generate a big data set, we said we want to be, as an AI company, you're a data company, and it's good to be data independent. So how can we generate that data set? So we set out to set up some naive synthetic libraries by using the, again, machine learning and observable antibody space. So we came up with 2 designs and get it, synthesized and then built and all of that. And we are using those libraries and off-the-shelf antigens and do screening in our lab in Cardiff to build a big data set. We are adding a number of projects every month and make that data set bigger and bigger. This year alone, we have done 46 projects. And in certain cases, to get more diversity, not on the antigen side, but also antibody side, we work with Global Bio. They kind of offered us 4 of their libraries, and we are using those libraries to have a more diverse data set. Briefly on the validation of the recovery module and the generating module. So the recovery part returns 6 to 13 times more cluster on multiple different projects. And the generating module show that we can actually design antibody sequences that are outside of the library. Give you some options that you haven't even thought before. And we show both of these in the wet lab that these are, binders. One of the latest case studies we had, this is with the top 20 pharma, they started with a human target and a murine homologue, and the plan was to do different type of panning arms and looking at the data and to pick for cross-reactive sequences. So they have a very high throughput way of screening the colonies. They do not just do a few hundreds, they do thousands, right? But we still manage by using NGS and our algorithm to find many, many more clusters and diverse sequences with a few counts in the whole population. And those sequences turn out to be binders. We gave them about 200 to 300 sequences, and 94% of them turn out to be positive binders. They were super impressed with that, and now we are following up that project with another one. And I should mention that, interestingly enough, within those additional sequences, we identified 5 cross-reactive sequences that they are really happy with. Another project we did, I won't get into too much detail here, is again looking for the enrichment profile. So how does our machine learning algorithm picks one sequence over another, or how does it do the clustering? So one of the ways how it does the clustering is, it is not only looking for the count of sequences from one pen to another, it is treating it as a population. So it's not only looking for that sequence, it is also looking for similar sequences and how that they perform from one pen to another. We call it as its enrichment profile. So did it get more and then less? How did it interact in the whole population? And the second, for each sequence, we create a representation of that antibody in a multidimensional space. We looked at that antibody from the perspective of sequence, from the perspective of charge, from the perspective of hydrophobicity, from the perspective of 3D structures and epitopes and CDR regions, and etc. And then we unified that, and then we clustered those sequences in this multidimensional space. So not so surprisingly, you find out that very similar sequences ending up in different clusters, and different sequences also may end up being in the same cluster, showing that actually machine is able to be determined at least a correlation that is not based on the sequence only, and can tie this group of sequences into an epitope. Another thing that we have done, this is not something we do very often, but what we did here, it was against TNF. We have been asked, look, here are the sequences we found against it. We would like to do something more interesting. Can you showcase that you can actually generate sequences that are not part of the library? And we designed about 24 of them. They selected 8 and showed that one of them actually binder. So when we analyze the fetch display, the success rate is about 94%. And when you are designing something almost from scratch, then the success rate is about 15%. But it is still interesting in a way if that is something needed for that particular project. I briefly will go back to the maybe third slide and tie it to here. So not only we offer this service, but we have a business model where we do a drug discovery project on difficult targets, mostly GPCRs. And it is a very lean process. In 6 months' time, we provide some functional antibodies, and in return, we charge an upfront fee, and then we have these headless contracts where the downstream terms are to be discussed later on once you see our data package. And we'll give you about 3 months to do the in-house validation and further testing before you decide to whether take license on the antibodies we designed or not. So these are the 5 that we are developing biologics for internally. And of course, we are in a few conversations with different pharmaceutical companies to either work on these together or work on new GPCRs. This is a fraction of our team with additional advisors Jonathan Milner, some of them might know. We have Jane Dancer, Justin Briggs, Kirsten Papanfus, Gary C. Hussain, a lot of very high caliber names in drug discovery and GPCR specifically. And I think with that, I will conclude my presentation. So if anybody would like to hear more or get in touch, I'm always open for a chat. Thank you.
Thank you so much for that, Murat. Very, very interesting stuff. You know, we have a lot of companies and individuals come talking about AI, of course, as probably the predominant technological disruptor of this space, but it's rather rare that we see such interesting work going on with AR. And it couldn't be at a more timely moment, I would say, given the you know, global shape of that vertical and how present it is in the public's eye lately.